Yeah, it's oh god,
I don't remember what its name was.
synthetic, artificial synthetic.
It's just,
Uh,
it was create a
con-
acidic bond.
Like a control was no, it's part of the control, right?
What is ONPG? It's an indicator, but what is it? It's a dimer. Excellent. It's very similar to lactose, except for it has an R group attached to it. When this bond breaks, it does what? Turns yellow. Excellent. So it indicates what is working. The enzyme. If it turns yellow, enzyme is working. Does it tell you how fast or how well it's working? No. All it tells you it is working. Okay, if it does not tell you, if it does not turn yellow, it is not?
It's not working.
Working. If I put this enzyme in an extreme acid, what happens? It denatures. Excellent. And then it will not turn
Yellow.
yellow. If I put it in an extreme salt solution, it'll do what?
It'll
Denature. What does denaturing mean?
breaking all structures.
Breaking structures. Typically it means you break.
The primary.
No.
Oh.
Secondary structure. In those
Secondary
cases, hydrogen bonding will get disrupted. Ionic bonding will get disrupted. Now, if I put it in extreme heat, what can happen? You could theoretically break some of those primary structures. Okay,
Yeah.
it takes a lot to break the peptide bonds, but you can do it. It takes more. Okay, what happens if I put it in extreme cold? Why does it slow down?
Because the molecules aren't moving that fast.
There you go. Molecules just aren't moving that quickly. If I leave ONPG on the desk for an hour, what happens to it? Talk to Sherry. If I leave it on the desk for an hour, what happens to it?
A couple of bonds will break.
Yeah.
If I leave it on the desk for an hour, what happens?
Some of the bonds will break.
Excellent. It will start turning yellow. Because?
You're breaking the glycosidic linkage.
But why?
All reactions are
All
spontaneous.
reactions are spontaneous. Yes. Okay. 5 years from now, those are the names you'll remember, and you'll pass some classes because of that. We'll figure that out. Okay. Substrate comes in. It fits the active site. It binds. It breaks apart. Enzyme works. Or we can assemble it. Enzymes can go. Reactions can go in either direction. We assemble, or we can break down. Let's imagine I have both ONPG and lactose present. What happens to the rate of the reaction? Does the reaction slow down? No. The reaction speed still is occurring the same, but the amount of yellow is slower for what reason? Competition. The enzyme does not know if it's ONPG or lactose, so it's just breaking that glycosidic bond. It's like, hell yeah, it's glycosidic bonds, let's break it. So it'll appear less yellow because the enzyme is doing what? Also breaking lactose.
It's
That's
breaking two
competition.
substrates at the same time.
Competitive inhibition. You can either activate an enzyme or you can inhibit an enzyme. You can inhibit an enzyme or activate it in multiple ways. One, you can change the active site. Two, you can change somewhere else on the enzyme that changes said shape of active site. If it's somewhere else on the enzyme, we call that what? Allosteric. Excellent. Nice job. Okay? So, what can happen here? Above my clip art, what can happen here? Talk to sure.
competition to see which one gets in there.
I actually didn't. That's a crazy condition.
Not all fit there, right?
They can all fit there.
What is this example of? Active site competition. So do we have an example of this that we did in lab? What is it?
The lactate.
ONPG and
The lactate.
lactose. Okay? This is ONPG, this is lactose. Does the enzyme know which one it is? No, both fit. Okay? Cofactors and coenzymes. Often enzymes require a second enzyme to work. Okay, two things come together to help catalyze the reaction. Or you have things that come in and fit to have things work. Allosteric means it binds somewhere not in the active site. Okay, so that is not the active site. But what it's doing is changing the shape. The enzyme, when just there, is this shape. When it comes in right here and binds, all of a sudden it's now this shape. That is activating it. You can inhibit it by, let's say this is the normal shape, and this fits the molecule really well. Something comes in right here and it changes it to this shape. Now can that thing fit in it? No, that's allosteric inhibition. Okay? Talk to each other. What is going on with each of those?
Okay, so I didn't understand. Inhibition, it doesn't fit. Is that what I understand, or am I wrong?
For which one?
Inhibition.
Yeah, so inhibition, I think both change the shape. Inhibition changes it where it doesn't work.
Oh, okay.
Activation changes it where it does work.
Okay, so both change the shape, but it doesn't work.
What is these molecules? Could be proteins, could be enzymes, could be ATP, could be anything. When it binds, it does what to the enzyme? Changes its shape. Therefore, changes its function. Shape equals function. So if I change the shape and now it can fit, it can now work. If I change the shape and it no longer can fit, it now
Cannot
no
work.
longer works. Activation versus inhibition. Not all enzymes require activation or inhibition. Okay? Some do, some don't. The lactase enzyme, did it require anything?
No.
No. It just works on its own. Okay, we're not regulating the lactase enzyme in that manner. In 10 more seconds. Talk to Trey.
I got a allosteric inhibitor.
I'm sorry?
I got A.
A. Because it's reducing the ability to bind, which means it's not working.
We're binding in the active site or outside the active site?
Outside the
Therefore, we call that what?
Allosteric.
Allosteric. So therefore, we're allosteric, and are we increasing or decreasing? Decreasing, so it's what?
An inhibitor.
Closing up. Go to the next question. Going. Seconds. All right, talk to Chair.
I picked ionic and hydrogen.
Yeah, 1 and 4.
I just picked ionic, but like I feel like it can also be D.
Yeah, 1 and 4. Oh, so yeah, A and D. I was looking over there, 1 and 4.
I feel like it's because of 1 and 4. But
Maybe not.
like I don't feel like it can. It's not like always hydrogen bonding.
Okay, yeah. I was.
Is it, if it's linear negatively charged, is that polar?
Yes.
It is polar.
Yes. Polar is anything that's not essentially covalently bound. So it is not nonpolar, and it's not going to be hydrophobic interaction because it's not carbon and hydrogens. Therefore, it is going to be ionic bonding. And if you can do ionic bonding, really you can do also
Hydrogen bonding.
Next question.
Choose. I don't know.
Okay, 10 seconds.
Oh, jeez. I was clicking on the.
I got 1, 2, and 3.
I said 1,
I
2,
just-
and 3 because they're all capable of ionic and H bonding. And I'm not sure about 4. I didn't click it because I didn't notice that there was a sulfur in it.
I don't know. I just put 3. But
Sulfur is.
Yeah.
That would be slightly overturned.
All right, first question. Which amino acid is this? That was the one that's universal that we're always going to start with.
I mean,
Methionine.
oh,
method.
That is methionine. Okay, so that's the most important amino acid because we always start with met. Can met? Would you like to find it there?
No.
No.
Right? Would you like to find this one?
Yes.
No, because it's negatively charged. So we need something that's positively charged. Therefore, it is going to be that one and
Yeah. I was wrong on this one too.
okay, close enough. Go into a new session. Oh, shoot. I'm on the wrong one, sorry. That one.
Bless you.
Thank you.
Fosforfrukt.
Okay. Talk to each other.
I put A, I don't know, but I don't know.
I think it's either A or B because the ATP is negatively charged and argine has a positive charge.
Okay, I'm going to give you a hint. It's not A.
Which makes sense. Because I clicked A and I was like, that sounds right.
That's-
I read it and I was like, okay. And then I read the second and I was like, maybe it's that.
So did you do V at the end?
Phosphates are negatively or positively charged?
Negative.
Negatively charged. Therefore, the correct answer is C.
Oh,
I would like to let you guys know this is your first MCAT question that you have now taken.
first what?
MCAT question that you've now taken. So there's your answers. There's an answer with my explanation.
Oh.
What?
For those that are getting ready to want to take your MCATs.
Oh, what is an MCAT?
MCATs are what you need to take to become a doctor. There's your entrance exam.
Oh, okay. like,
Medical doctor.
like I'm lost.
That is an MCAT. You want to take your GREs? GREs are very similar,
I have to take
but it's for any graduate school entrance exams.
take a picture. Take a picture.
Take a picture. Okay.
Wait, oh, I know. I got it. I got you. Wait, I got you.
I missed.
I think that makes sense though.
Can you go back and
He was
access
saying
them?
negative charges repel each other, which makes sense.
Yeah.
When you exit.
Oh, repel. Oh, that makes sense.
I did A and C and was unsure about E.
I did A and E, I think. But I don't know. I didn't know what I was looking
I
for.
was unsure about E because like 105 is a temperature human bodies can reach.
Yeah.
But does that stop like a ton of enzyme reactions? So that's
I have no idea. So which ones you picked?
I did A and C.
A and C.
What did you do?
A and E. So you said
I said C was true because at pH 2 and temperature of 98 it would have the highest.
but says it doesn't talk about temperature, or is it talking about temperature?
It talks about the pH of 2, which is true on the first graph, and a pH and a temperature of 98,
And on A, yeah.
which would be true for the second graph. So that's why I picked C.
Oh.
Pepsin's heat reactivity will be around a pH of 2. Excellent. The speed of the human enzyme increases in the environment from 70 to 108 or so, right? What happens when you have around 100? Starts decreasing. So when you have an extreme fever, what happens to your enzymes?
They slow down.
They stop working, right? To slow down trypsin's rate of reaction after the pH of the surrounding environment goes from 8 to 9. Does that sound good?
Yep.
So B, D, and
B, D, and E. Well, I got one.
I don't think it is C. Because it doesn't stop at 100.
I don't know. We all have different orders. They're all different orders.
All right, top sure.
I picked B. Oh, it looks like it
I said C.
I did too,
Yeah.
but I don't know.
Because A is about temperature, B and D are about concentration, and C is about structure
We're
alteration.
going to lose our tertiary structure.
Oh.
Okay?
Okay. Whatever you say.
What happens with pH changes?
You get more hydroxides.
But what? What's happening with what is an acid? What's a base? Protons
Oh, yeah,
or
yeah.
lack of protons, right?
Yeah, that makes sense.
So that's going to affect what type of bonding? Hydrogen bonding
H bonding.
Yeah.
within the tertiary structure.
Yeah, that makes sense.
Correct.
Of course.
Our goals today is to understand membranes. Membranes are really important because that's what makes up you. These are our typical lipids. Fats, triglycerides, phospholipids are what make up your membranes. So phospholipids are just a long hydrophobic tail with a phosphate group or a polar end. So you have a polar side and a hydrophobic side. That allows you to have basically a water side and an interior side. And then steroids. Steroids are large hydrocarbon chains that allow to be able to pass through the membranes or to be embedded in membranes. So when you have like high cholesterol, that's going to change the fluidity of your membranes. You want a balance of having somewhat fluid versus somewhat rigid membranes. So we're going to control that by the length of the tails and the amount of cholesterol present. Triglycerides, simple formation, this is a redox reaction. We're going to take these large hydrocarbon chains and we're going to assemble them through the carboxyl groups. Very similar within the large hydrocarbon chains, which we did talk about earlier. Saturated versus unsaturated, trans versus cis, conformations. Those are your fats and lipids. All right, let's start applying it. Very good.
Okay. Got this.
All right, Dr. Jordan.
A.
I said B.
A's got it's polysaturated, so it's got more saturation, I think,
I think
because of the double bonds.
so. Saturation is more H's, so that's less double bonds. Unsaturated is when there's non-optimal hydrogen. I said B because it had
Less.
double bonds, and it also had a shape where I thought they could be closer together. Like if we had more of the B's, they could be really close together, which I feel like would be more rigid.
Okay. Yes.
Yeah, saturation is, are there as many hydrogens as possible?
Yes.
So the double bonds are when there's not as many hydrogens as possible.
Yes. So you're saying the more H's, the more permeable?
The more H's, the more saturated, which is the more rigid it is.
No.
So more saturated, less fluid.
Which one's going to have the most fluidity? A. Excellent. A's going to have the most fluidity. It's going to be able to, because it's not saturated. It's long, not saturated, and it's kinked. So you're not going to be able to get a whole bunch of them just lined up right next to each other. What's the next most fluidity? C, for the same reason. But B is going to be the least fluid. Okay?
Just like you said. I was taking it backwards, as usual.
Yeah, I think I was confused about that last night too. Water.
Is it the outside or the inside?
Well, the green parts are hydrophilic. The red parts are the hydrophobic carbon sheets.
But what's better, to be on the inside or in the outside?
It's either A or B. I think, because in the reading they were talking about when it's just surrounded by water, that's unstable. But like separating the water makes it spontaneous. But
The green dots are.
I don't know.
Oh.
What's this represent?
Wait, what?
Hydrophilic.
Hydrophilic, hydrophobic. Okay? So we're going to have a hydrophilic side, hydrophobic middle, hydrophilic side. So it's going to be B.
All right.
And we're going to go in perpetuity, therefore you're going to create a sphere. Because if you go in all axes, so you're going to have an axis go this direction, and you're going to go in this direction, that creates a sphere. What's a cell?
Sphere.
Sphere. Has a lipid membrane. How does soap work? It's a good question, right? OChem should know this. How does soap work?
Wasn't it?
I'm sure you had that on the exam at some point.
Oh, we talked about it 2 times, and I just remember that it
What do they teach you over there?
Disrupts hydrophobic
was
interaction.
hydrophobic, wasn't it?
All right.
It was,
What
but the outside.
is dirt? Okay.
Tierra.
You have to go do stuff on your car. You changed your oil. By the way, learn to change your oil. So much cheaper, it's better for the world. Just go learn to change your oil.
For the world.
$150 when you go to Costco, buy $30 for 2 packs of oil, $5 for an oil filter. Like, learn to do it, okay? It literally takes 5 minutes. The worst cars in the world are Volkswagen, and Volkswagen takes like 15 minutes, okay? Literally, they're the worst cars. So your hands are all dirty. What's that dirt? What's the oils? Lipids, it's fats. It's stuff that's going to be hydrophobic. So when you just pour water on it, what happens to it? Nothing. It flows off. So what are you going to put on your hands? Soap. What is soap? Has hydrophilic end and a hydrophobic end. So the hydrophobic end is going to grab onto the oils. And then it's going to connect to the hydrophilic head, and it's going to wash it away.
Goes like.
That's more what C is and A. A, typically one end, creates micelles. Micelles are what trap things inside of them. Okay? Yeah. They do the same thing. Yeah. Okay? So, that's soap's really important. Let's get started on the worksheet. I need to leave for about 5-10 minutes. They hadn't finished printing your pre-exam, so I got to walk over and grab them. So I'm going to go do that. Take a break. Start working on the worksheet. We're going to come back to you. Yes.